A very good morning to everyone. Welcome in the class. And as we say, let's crack CSIR UGC NET together. We are doing this topic under the category of GATE crash course. I have already made a video on RDNA technology enzymes or the modification enzymes needed for RDNA technology. Part 1. Some of the enzymes which could not be included in that first video, we are going to just continue from there, and you can watch the previous video for the rest of the enzyme, and this video will be covering 3 enzymes, right? So before moving ahead, let us have a small introduction about myself and Unacademy. Unacademy is one of the India's largest platform for the preparation of CSIR UGC NET. I'm your educator Farida Johar Bandukwala. I have teaching experience of 10 years. I've qualified MP SLET examination in year 2018. Unacademy, if you're choosing an academy for your preparation, then what all things you are going to get? You're going to get daily life classes. You're going to get the syllabus in a well structured manner because you have to complete the whole syllabus in a small span of time. Then live tests and quizzes are taken so that you can evaluate yourself that how much you have learned so far, and then whichever category you are going for, you will get unlimited access towards all the videos in that particular category. Unacademy tries to give free live classes under following thing, that is YouTube and special classes. So for YouTube, you just need to subscribe the channel, and the name of our channel is Let's Crack CSIR UGC NET. And for special classes, you need to download the Unacademy Learning app. After downloading the app, make your account over there and you'll be asked to fill the referral code of an educator. So use my referral code FP01 so that you'll be able to attend the free live classes of mine as well as other educators. So go for the plus subscription. Plus subscription is a paid subscription, but all the things in the syllabus would be covered in the plus subscription for all the upcoming classes. Various crash courses and short-term courses are conducted. Then you can get the whole study material in PDF format. So just go for the plus subscription and use my referral code FB01 so that you will be getting an additional 10% discount. So do not forget to use my referral code FP01 for getting the plus subscription. Okay, so let us move ahead and let us talk about the enzymes required for our DNA technology. We have talked about nucleases, we have talked about polymerases, and we have talked about restriction enzyme in our previous video. Because of a certain technical issue, the video could not be completed over there. So here we'll be talking about 3 more enzymes, that is ligases, methylases, and topoisomerases. So in this video, we'll be discussing 3, and 3 I have discussed in the part 1. So let us talk about ligases. So ligases are the enzymes which are responsible for joining the nucleic acid molecules together. Let me just put my specs. So ligases can join the nucleic acid molecule, can join nucleic acid molecules together. They are able to join DNA as well as RNA, can join DNA and RNA. So it is DNA ligase and RNA ligase. Both of the types of ligases are there. After that, DNA ligase, it catalyzes the formation of phosphodiester bond between the 5' phosphate of one strand and 3' hydroxyl of the another strand. So catalyzes formation of phosphodiester bond. So ligation in the form of phosphodiester bond between 5' phosphate of one strand and 3' hydroxyl of another strand. So most of the DNA ligases which we use are actually derived from bacterium, page P4. Most DNA ligases Most DNA ligases are derived from bacteriophage T4. Okay, so this is the thing. And another thing, DNA ligases, they are also called as molecular glue. Like restriction enzyme, they are called as molecular scissor. Ligases are called as molecular glue because they are responsible for sticking the things. Now, as we have seen that what is the function of restriction enzyme normally in nature, why nature evolved the restriction enzyme? Because of the attack of bacteriophage, the bacteria needs to save itself from the attack of bacteriophages. It developed the resistance mechanism to overcome the infection of bacteriophage. It developed the restriction enzyme. So what is the biological function of ligase? Why was this ligase evolved? So ligase evolved to perform the repair mechanism of single stranded breaks or nicks. Single stranded breaks or nicks. This is the function of ligase. Now, ligases can perform this sticking in two forms, that is like restriction enzyme, it produces blunt ends and it produces sticky ends. So same with ligases, they can stick the blunt ends or they can stick the sticky ends. So blunt end and sticky ended ligation. Blunt ended and sticky ended ligation. So ligation reactions may be blunt-ended or sticky-ended. May be blunt-ended or sticky-ended. First thing. Second thing, sticky end increases the efficiency of ligation because already there will be a complementarity. So complementarity is going to bring them together. So sticky ends has high efficiency towards the ligation. So another thing, sticky ends increases the efficiency of ligation. Right, so blunt ended or sticky ended ligation, we have already seen blunt ends. What are the blunt ends? Like this, the blunt ends would be like this, and the sticky ends would be like this. There will be lots of overhangs, right? So blunt end and sticky ends. Now, after that, let us talk about the types of DNA ligases. Types of DNA ligases. So there are two families of DNA ligases, and what are these two families? First is ATP dependent and second one is NAD dependent. These two are the families of DNA ligases. If we are saying ATP dependent, then they are found in eukaryotes, found in eukaryotes, and NAD dependent, they are found in prokaryotes, found in prokaryotes. So these are the two families. Now in mammals, there are four types of DNA ligases seen. So they are type 1, type 2, type 3 and type 4. And if you remember that the types of restriction enzymes that we have seen is of three types, type 1, type 2 and type 3. So in mammals, there are four types of DNA ligases which are seen. Do not forget this thing as well. Now talking about methylases. Methylases. These methylases are also known as methyl transferase, also that you can understand the function in a much clearer way, also known as methyl transferase. Methyl transferase. Now these methyl transferase, where are they found actually? So they are found in bacteria till mammals. So they are found almost in everything. The range is vast. Found in bacteria to mammals. Right? This is about methylases. So usually the organisms that make the restriction enzymes, they also synthesize methylases. Why? Because these methylases are now going to protect the DNA from cleavage, because every time the sequence for which they are specific, like for example, they are specific for a certain sequence which is found in the virus, and the same sequence it has also got. So how is it going to save itself from its own restriction enzyme? So for that function, they have got methylases to protect its own DNA. Understanding? So let us write this and then we'll understand this. So organisms that make restriction enzymes, restriction enzymes also make DNA methyl transferases. DNA methyl transferases. Why? To protect its own DNA from cleavage. To protect its own DNA from cleavage of its own restriction enzyme from cleavage. So as we see the normal function, why the particular enzyme evolved in nature. So methylases, it evolved in nature to protect its own DNA from its own restriction enzyme. If the sequence for which it is specific, the restriction enzyme, that same sequence it has also got, right? So methylases. Now these enzymes, they recognize the same DNA sequence as the restriction enzymes they accompany. So recognizes same DNA sequence as restriction enzyme they accompany, right? After that, methylation. What is methylation? Basically we are saying methylases, so what it performs. So methylation is a process of addition of methyl group or adenine group or cytosine base within the recognition site, and thus modifying the site and prevent the action of various restriction endonucleases. So we'll write this. What is this methylation? Methylation is addition of what all? Methylation is addition of methyl groups to adenine or cytosine bases. Cytosine bases. And where? Within recognition sites only. Within recognition sites. And thus modifying and thus modifying the site and prevent DNA restriction. So addition of methyl group to adenine or cytosine bases. Right. Now types of methyl transferases in mammals are of four types. So we'll be seeing that four types of methyl transferases in mammals. Methyl transferases in mammals. So first, second, third and fourth. First is DNMT1. Second is DNA methyl transferase is type 2. Third is DNA methyl transferase 3A and DNA methyl transferase 3B, and this 3B is also called as de novo methylases. Remember this, de novo methylases. Then the fourth one is DNA methyl transferase 3A. So methylation, this process of methylation affects the restriction. Restriction enzyme will generally not cut the molecules where a particular base within the recognition site is methylated. So let us write that its function. So function, methylation affects restriction. Now, why methylation affects restriction? Because restriction enzyme will generally not cut molecules where particular bases where particular bases within recognition site are are methylated. So this is the function of methylation, that it affects the restriction. After that, we are going to talk about topoisomerase. Topoisomerase, the next enzyme in the category of the modification enzymes needed for genetic engineering or RNA technology, is topoisomerase. So, topoisomerase, something related to the topology of DNA, that it does not change the basic composition, but it changes the structure. That is the meaning of the term topology and isomerase. Like the chemical structure of fructose and glucose is same, but they differ in their structure. They differ in their functional group. So topoisomerase, they are going to change the topology of DNA. So how they are going to do that? That we are going to see. They regulate overbinding or underbinding of DNA. They regulate overbinding or underbinding of DNA. Now, where are they found? First of all, their function. So they are going to regulate if extraordinarily they are bounded or underbound. Those all things will be taken care by topoisomerase. Now, where are they found in? They are found in vast number of cells from viruses to mammals. Found in viruses to mammals. Right? Now, if they are doing this going to regulate the overbinding and underbinding, so how are they going to do that? So they make incision in the DNA backbone. That is what they do. They make incision or a cut, incision or cut, incision or a cut in the DNA backbone. That is the phosphodiester bond, DNA backbone. And they can be classified into two groups based on the number of strands they break, whether they are able to break one strand, single strand, or whether they are able to break both of these strands. So can be classified into two groups. Classified into two groups. What are these groups into which they are classified? They are classified into type 1 and they are classified into type 2. Right? These two are the groups. Now type 1, it is going to make a nick in one strand and passes the intact strand through the nick and reseals the gap. Resealing the gap is also done. Type 2 is going to make a double-stranded break, create a gate through which the second segment of the helix is passed. So that now the torsion is. So let us write the function of type 1. Makes nick in one strand, makes nick in one strand and passes the intact strand, the intact strand to nick and reseals the gap and reseals the gap. This is about the type 1. Now talking about type 2, makes double-stranded break and creates a gap to which a second segment of helix is passed. Right? This is about our topoisomerase. Now, if we are talking about topoisomerase, we should also know, we have seen that what is its basic function. Type 1, remember that type 1 is responsible for making a single stranded break and type 2 is responsible to make a double stranded break, right? Now, when we are talking about this, we need to know the mechanism as well. So, we'll be talking about the mechanism of action of topoisomerase. So, mechanism. So how it is going to do this? Rotating the broken strand around the intact strand to relax the strain on the DNA helix, followed by resealing of the broken strand. As easy as this. So let us draw a structure for this and then we'll be talking about the theory. I'll be writing the points. Okay. If this is your DNA, right? So we are talking about the type 1. Here we'll be drawing for type 1. See, and this is how, and here, and like this. So it has made this nick over here. This is the nick, and now the structure is going to become like this. This is how it has released the strain by type 1. Now talking about type 2, that how it performs this. Let us make a double-stranded DNA. This is our double-stranded DNA. It will perform the cuts in both of these strands like this. After that, it is going to rotate the strand and reseal them. Reseal them. This is how the type 2 performs. So let us see this in the theoretical way. So rotating the broken strand, rotating the broken strand around the intact strand, around the intact strand. So the broken strand will be rotated around the intact strand to relax or to unwind, relax or unwind the strain of DNA helix, to relax the strain on DNA helix, followed by resealing of the broken strand, followed by resealing the ends of broken strand, broken strand. First thing in the mechanism. After that, cleavage by the topoisomerase results in the 5-hydroxyl and 3-phosphate termini. You should not at all forget this sentence. I'll be writing it in caps. Cleavage by topoisomerase results in 5-hydroxyl and 3-phosphate termini. Right, this is the thing. Do not at all forget this, because this is an exception. Usually 5- is associated with phosphate, but here it is associated with hydroxyl, and 3- is associated with the phosphate. So DNA topoisomerase therefore performs the function of cutting as well as joining. So nuclease and ligase activity, both of them. So DNA topoisomerase therefore has both nuclease and ligase activity. Okay. After that, DNA topoisomerase, they are involved in the processes that require turns of the double helix to be removed or added to the double-stranded molecule. So, DNA topoisomerase are involved in the processes which require the turn of the double helix to remove or add the double-stranded molecule. After that, they achieve this function by causing a transient single or double-stranded breakage in the DNA backbone. So that we have seen to relieve, strain, transient cuts are made. Transient cuts are made. Right? So this is the function of our DNA topoisomerase. Now the last enzyme that should not be forgotten at all whenever we are talking about the modification enzyme, that is DNA gyrase, and that is the last enzyme, DNA gyrase. So DNA gyrase is actually a type of topoisomerase enzyme which belong to the category of type 2. So DNA gyrase is one of the types of topoisomerase enzyme called as type 2 topoisomerase. Then DNA gyrase is responsible to relieve the strain, right, as a function of isomerase, relieves the strain, while double-stranded DNA is being unwound by the helicase, relieves the strain, and double-stranded DNA is being unwound by helicase. Now, this is the introduction about DNA gyrase, and we always see the mechanism of action, that how it is going to do. So the next thing that we need to see is mechanism of action. Mechanism of action of DNA gyrase. So how it performs this? So gyrase is going to bind to the DNA. Gyrase binds DNA. So this binding to DNA is called as gyrase DNA state. Gyrase DNA state. So there is a competition between DNA unwrapping and dissociation. There is a competition, while it binds, there is a competition between DNA unwrapping and dissociation. and where increasing DNA tension increases the probability of dissociation. So we'll write this in another page, that there is a competition, there is a competition between DNA wrapping and dissociation. DNA wrapping and dissociation. So whenever DNA tension increases, when DNA tension increases, the probability of increases. So whenever DNA tension increases, it increases the probability of dissociation. Increases probability of dissociation. So upon unwrapping and ATP hydrolysis, two negative supercoils are introduced into the template, providing the opportunity for subsequent unwrapping and supercoiling events. So what are we saying? That upon unwrapping of DNA, upon unwrapping and then introducing a cut, that is ATP hydrolysis, two negative supercoils are introduced. Two negative supercoils are introduced into the template, are introduced into template. And when this happens, it gives the opportunity for the subsequent wrapping and supercoiling event, which leads to wrapping and supercoiling, which leads to wrapping and supercoiling. So in this whole process, two molecules are hydrolyzed per cycle of the reaction of a gyrase. So this we'll write in another words. So two ATP molecules are hydrolyzed per cycle of gyrase reaction, gyrase reaction, and leading to the introduction of a linking difference of -2, leads to linking difference of -2. Very important point to remember. When we'll be doing the calculation of molecular biology, we'll be talking about the linking number and everything in detail. So like this, we have covered the enzymes in detail. In previous part 1, we have done the nucleases, we have done the restriction enzyme, and we have done the polymerases. In this video, we have talked about the fourth enzyme, that is the ligase, then methylase, then topoisomerase, and lastly gyrase. So like this, we have covered 7 enzymes in the category of modification enzymes needed for RNA technology. So this is it for the class. We'll be meeting again tomorrow at 10:30 a.m. with a topic of CSIR syllabus, and in the evening at 5 o'clock with the syllabus of GATE Biotechnology 2021. As we say in the end, let's crack CSIR UGC NET and GATE 2021 together. 2021, and that too for biotechnology. Biotechnology. I hope you people have understood the things, and if you really understood and you like the video, give it a thumbs up and hit the bell icon for getting all the notification of the upcoming videos. For attending the plus classes, you need to get the subscription. So for getting the subscription, use my referral code FB01 and you will be getting a 10% discount. And even for attending the free special classes, free special classes, use my referral code FB01. By using my referral code FB01, you will be able to attend my special classes as well as the special classes of other educators. So I hope you have understood the enzymes needed for RDNA technology quite well. In another class, maybe after some days, we'll be talking about the whole process of recombinant DNA technology. So thank you very much for your patient listening. Till we meet next time, keep on studying and be happy. So this was it for the class of today. I hope to see you in the other classes as well. Finally, it's a bye from my side.